So welcome, Dr. Todd Hoare, to our Expert Corner interview. Thank you for joining us.
My pleasure.
So let's begin. So how does a small town boy from Tory Hill, Ontario, population of 200 people, end up in this field?
That's a great question. Not a lot of us end up doing this pathway from small towns. I think that's something that I really wish I'd see more of. But yeah, absolutely. So I decided to go to undergrad for engineering at Queen's, and I think I picked Queen's because Kingston was the smallest town I could possibly find with a decent university. I toured U of T, but coming from a 200-person hometown, that was a little bit intimidating. So I ended up doing that with engineering chemistry, and along the way, I sort of got different jobs that intrigued me around the area of polymer science. So my first job was actually doing asphalt research. So we were taking different bitumens and putting polymers in them to try to prevent cracking in the winter, right? In Canada, it's a huge problem.
It is.
So I think I ruined many pairs of pants and shirts in that process. And I eventually was walking to work basically covered in asphalt, but you know, I must have looked homeless all summer, but that's okay. Yeah, but I think it involved a lot of mechanical property testing, which I thought was interesting. And it also showed me how polymers could be used in different applications to improve their properties. And that sort of was a really point of interest for me. So I ended up doing a summer at Xerox making toner, which was brought me into sort of the polymer particle world, which led me to apply for graduate positions in colloid science and polymer colloids, which led me to McMaster working with Bob Pelton. And that brought me into the microgel world. So using polymer particles, but now particles that were gels. And that really intrigued me. And I was really interested in the idea of how to customize the properties of hydrogels and use those properties. Initially, it wasn't even an application-driven thing. It was just fundamental physical chemistry. But then I ended up making a glucose-responsive gel, and the applications for things like insulin delivery were kind of obvious. So that sort of pushed me into drug delivery, and then in my postdoc, I really expanded on that, and I spent about 8 months doing neurobehavioral testing on rats, handling them 7-8 hours a day, having them poop on me, run away, which I'm not sure I would want to repeat necessarily, but I think again, just seeing the materials design, where it was sort of my training, through to that application of seeing how something worked in vivo, sort of how I've built my research program around that model to sort of really understand the materials and how to synthesize the material and what the properties of that material might be, but then really think about application performance and can we actually use this in a practical context? And if we were to use this idea in a practical context, what are sort of the barriers that we'd have to think about to get there? So it's sort of a circuitous route, but we got there.
That's great. So so you sort of fell into this, or were there people that actually influenced you, or were there other influences basically that that helped you on this life science train?
I mean, I guess I'll give some credit to Bob Pelton, my PhD supervisor, because he basically just paid me and said, "Do whatever you want," which is kind of a not always what you get in graduate school, but so supporting whatever decision I made. So I mean, he is not a biology person at all, but I ended up doing some of the drug delivery work just because that's where my interests in that work happened to take me, and then sort of made decisions to sort of build on that. And so I think having people who sort of believe in you and sort of give you freedom and in research, it takes a bit of money too, right, to actually get there. That was really beneficial for me to sort of explore different areas and find out what I really was interested in and where I thought I could make a contribution.
Indeed. So beyond the studies and your impressive experience, we heard that you like to play piano and are an avid curling player. Can you tell us more about that?
Sure. Well, I took the Royal Conservatory of Music through the end of high school, so I did a lot of classical piano, but nobody cares about classical piano. So I actually Actually, I've played for, I think, 3 or 4 weddings, but they're all for friends. So one of my ambitions before I die is to actually be paid once for a musical gig, so I can call myself a professional musician. But I play every few weeks at my church I would hesitate to call it a rock band, but more contemporary band, and it's a good release. It's sort of using the other side of your brain, but at the same time, music is very structured too. I think there's a lot of engineers that play instruments, and I think it makes a lot of sense actually.
Yeah.
It's a very similar skill set in a way, sort of taking that structure, but also adding in the creative element.
Yeah.
And in curling, I don't think I ever said I was good at curling. I just said I curl. The same thing. If someone asks me if I golf, I say I have clubs. So I think that's the correct answer. But it's something again enjoyable. It's a group of professors actually that curl, and usually we end up getting whipped badly by the senior citizens who can barely bend down but curl 4 times a week, and also the junior girls varsity team. Hands it to us every time. So I think it's a very equal opportunity.
So a regular humble pill.
Yeah, you can say that. It is in curling, the tradition is that the losers buy the winners drinks. And let's just say I haven't invested a lot of money in this year's curling. So.
Got it. On the music piano, what kind of music do you like to play on the piano?
I do still like
popular or not. What do you enjoy to play?
I like whipping out the classical books every so often. It's just exercising your fingers in ways you don't have to when you're playing in a band. Yeah, and my son's taking piano too, so we've done a couple of duets and things like that, which is kind of fun awesome. to do with your kids. So
great. So as I mentioned earlier, we are going to be going through some of your background and some of the leisure aspects of what you do, but we're also going to be going and diving deep on the science side of your background, which is the whole point of this expert corner, is to learn more about your background, your experience, and the work that you're doing with you and your teams. So I have Demetria joining me here, or who has been with us. Please, Demetria, it's all yours.
Thanks, Kathleen. And first of all, thanks, Todd, to be here with us today. And so I would like to start asking about your lab. So how is the organization right now in your lab? Do you have master students, PhD, undergrad, postdocs? How it is now?
It's all of the above. So I have a team of, right now, I believe I have 3 postdocs, 16 grad students, and we have a huge number of undergrads usually in our lab. So I think right now it's about 13. Because I really believe that undergrad training, it takes a lot of work on our end, but I think the value of it is extremely high to get people exposed to research, understand what research is, even just from an educational perspective, to experience failure. Because a lot of these students that we have are, they have 98s in high school, they have A's in university, and research is a very humbling experience because a lot of it, often what you're trying to do doesn't work if you're trying to do something interesting and new. So I think the value of that training is very high. So we put a lot of effort into that in my group.
Oh, that is very nice. And among your students, which are the main research lines, if we can divide like that in your group?
Sure. So I would say all of our research is around hydrogels or cross-linked polymer networks. I think that would be a general theme. But we sort of spin out in many applications from that. So we do quite a bit of work in drug delivery. So trying to, particularly within what we call in situ gelling hydrogels. So things that if you've used epoxy glue, it's the same idea. Basically taking 2 reactive precursor polymers and mixing them to form a gel rapidly. And we think that's really beneficial for minimizing how invasive hydrogel therapy might be to deliver drugs locally, and also using microgels, so these small nanoscale or microscale drug particles that, depending on how big they are, we can turn them to circulate, we can tune them to shrink or swell in particular biological environments. So for example, in a tumor where you have a slightly different temperature, slightly different pH, a slightly different redox environment. Can we make these shrink, swell, degrade in those types of locations to deliver drug more effectively at those places? And also to sort of transverse biological barriers. So one of the neat things about particularly nanoscale gels is that they're really squishy, right? So particles that can't get through these barriers in many cases when they're hard. Now, when you have the same size of particle, but you make it really squishy, they can get through. So getting to the brain, for example, or getting to the back of the eye. These are really challenging places to get drugs. So we're doing a lot of work trying to use these very soft particles to get there. We do work in biosensing, which is basically trying to work with a series of collaborators who know about the sensing part that we don't know about, but trying to solve some of their issues around fouling, around increasing signal to noise ratios in biosensing, which is a huge challenge, particularly for these point of care biosensors, where ideally you'd be taking a sample directly from a patient, dropping it on a sensor and hoping to get a number very quickly. And so you have to deal with all these elements that are very difficult to deal with. We also do some work in agriculture, which sounds a little bit weird based on the other stuff, but it's really drug delivery just to plants. So there's a whole other host of other barriers you have to think about adhering to leaves, transferring through waxy layers, getting into plant circulation. How can we design materials to do that? And also in the area of anti-infective surfaces, which is, we were actually doing this before COVID. We're not complete opportunists, but it's sort of been accelerated by a lot of the concerns that we've had in the pandemic in terms of how to make sprayable coatings that are stable, that can actively fight off bacteria or biofilm information over extended periods of time. And I guess the last area we work in is tissue engineering. And we really, in that perspective, we're really trying to address some of the material challenges that we feel are present in existing tissue engineering matrices, and develop ways to streamline the production of tissue engineered matrices so that either they're, can be directly injected into the body, you don't have to surgically insert them, or that we can add cells and make the structures that are needed at the same time instead of doing one and then the other. So those are sort of the areas that we really focus on, but all of the sort of key technology is I view our lab as a materials lab. So it's really thinking about the hydrogel that we need, the polymers we need, how to assemble them, how to link them together, and then how that relates to the application we're targeting.
Okay, great. Thank you so much. So one of the applications you mentioned is tissue engineering, and you mentioned also that you are working to solve some challenges of the materials that are being used in this field. Could you tell us a bit more in details which challenges you are specifically working on to solve?
Sure. So as I said, one of the key barriers, we think at least, to some of these therapies is that typically a hydrogel, or whatever the scaffold is, it doesn't have to be a hydrogel, but it's made outside the body. And the reason that's typically done is that in order to get the porosity that you might need for tissue engineering, to get the cell distributions, all those types of things, it's very difficult to do that and still keep something injectable or minimally invasive deliverable, I guess, if that's the word. So what we're trying to do, or that's one of the key barriers we think to actually using a lot of these scaffolds in the clinic, because if it's a surgery versus if it's an injection, it's a huge barrier in access and doctor time and hospital resources. So we're trying to develop a lot of methods to create these scaffolds that we can inject them, or we can put cells in at the same time as we make these structures, to sort of streamline the entire production process. So minimal risks, or reducing the risk that something doesn't become sterile throughout the process, making it less invasive to apply, being able to add different types of cells in different locations in the scaffold to try to generate more complex tissues, which is kind of difficult to do if you make the scaffold and then try to load cells after the fact in different locations. It's very challenging. So those are the types of things that we've been really thinking about, trying to streamline the manufacturing process and thinking about how do we design materials that we can still get the structures we want, we can still have cell-friendly conditions, but we can make this a little bit easier to get into a patient.
Okay, thank you. And so which main biomaterials are you working on for the tissue engineering?
So we're taking a bit of a different approach. So most people work on, degradable polyesters like PLGA, polycaprolactone, or, things that are in the natural extracellular matrix, so collagen, elastin, or, carbohydrates that are sort of mimic those properties. So we believe there's a lot of value in that, and we do use those materials as additives to a lot of our scaffolds. But we're really focusing on synthetic polymers. And the reason that we've made that choice is that it sort of frees you from a lot of the barriers involved in using those conventional materials for hydrogels. Because if you're using a natural ECM material that's in the natural extracellular matrix, it has a biological function. And whether you want it to execute that biological function or not in your tissue implant, it's going to do that. It has those cell adhesive ligands. It has all those signals that the body is going to recognize. So we believe with the synthetic polymers, we have a lot more control over what the body is actually going to do with this implant. So if we want it to adhere to cells, we can modify it so that happens. If we want it to deliver a growth factor, we can modify it so that happens. But we're not sort of beholden to the material properties or the innate functions of those materials that are hard to avoid. It also gives us a lot more flexibility in the chemistry and how we assemble them. So the polymers we use most often, there's really two polymer types that we are particularly engaged in. The first is something called polyoligoethylene glycol methacrylate, which I will call PEGMA here on for obvious reasons. I don't want to keep saying that, but it's basically polyethylene glycol is widely used in tissue engineering applications. Very good cell repellent polymer, which isn't necessarily good for the cells inside, but it's excellent for preventing fibrosis and desirable things from happening on the outside. But it's also a polymer that only has two functional groups in every polymer. Just by the way it's made, it's very difficult to get more than two functional groups. So the way that we make this PEGMA-based polymers is the PEG chains are on the side, but the backbone is very easy to polymerize using free radical methods and other methods that are very easy to do. And as a result, it's really easy to add other functional groups in, which gives us a lot of flexibility on cross-linking, gives us a lot of flexibility on how we want to functionalize the gel to make it do the biology we want. And the other benefit of PEGMA is that depending on the number of those ethylene oxide repeat units we put on the chain, if it's really short, it's actually a thermoresponsive material. So it can actually contract at physiological temperature. That gives us higher cell adhesion. It also gives us the ability to do some smart responsive scaffold design. As we make it longer, it's very protein repellent and acts more like PEG. So it gives us a lot of flexibility in terms of what the scaffold or how the scaffold is going to interact with cells and its environment. And the other type of material we were doing a lot of work on are zwitterionic hydrogels. So these are hydrogels based on polymers that have both a positive and a negative charge in really close proximity in each repeat unit. And the reason we've been interested in those is that they're very good for preventing fibrosis. So for example, implantable cell delivery vehicles, or cell therapeutics rather, where the idea is that the cell is supposed to be inside the gel and pumping out some sort of therapeutic over time. So you can imagine an artificial pancreas, for example, is sort of the classic example. The key barrier to those working is that the body doesn't like foreign things and it likes to wall them off, right? So if that's happening, you don't get those glucose signals in and you don't get the insulin out. And then the material eventually becomes useless, and then sometimes very quickly. So the very high anti-fibrotic nature of those materials is really, really interesting to us. We've also found that even though you would think that cells wouldn't like it, if you put cells inside in 3D, they actually survive extremely well and much better than a lot of the other hydrogels that have been reported. So we're really interested in using this, less so for tissue engineering in terms of like adding cells and having the scaffold reformat native tissues, but more for delivering cells and also for making cell-based therapeutics where the cells can be stabilized inside a gel for an extended period of time.
Okay, and so for the tissue engineering perspective, are you focusing on a specific tissue, or these are just general hydrogels that you are working to work with cells in general?
So I don't consider myself an expert in any sort of disease. So it really depends on what our collaborators say they're interested in. So we sort of try to develop materials platforms that for a particular application, someone can come to us and say, okay, I need this cell encapsulated and it has to last this long. We can say, okay, we know how to do that, and we go and make that type of scaffold. So we're very agnostic on application. We're really more focused on developing a toolkit of materials that we know how to engineer to meet different needs, so then we can sort of plug into different applications as the opportunities arise.
Okay, I see. And on this point, what about the degradation? Is this something that you are able to tune, or how you are studying this in your hydrogels?
So that's a great question. So again, a synthetic polymer, usually that's a concern, right? So you've got sort of these carbon-carbon bonds in the backbone. There's no way for the body to degrade that naturally. So, but it's also a concern with natural polymer-based hydrogels because you have very little control over that degradation process. Whatever polymer you use, you're sort of, again, beholden to the native oxidative or enzymatic or hydrolytic environment of that implant. So the way that we approach this is that we make synthetic polymers, but we make them very short. So we keep the molecular weight below 30,000. So that's sort of the lower limit of what people will say the kidney can clear. It's between 30 to 60,000, depending what paper you read. And then we link those small polymers together with bonds that can degrade. And the benefit there is that depending which bonds we choose, we can control the degradation time of our material. So the hydrazone bonds are the most common chemistry we use. And in neutral conditions, that's usually, a few weeks type degradation. In acidic conditions, it's faster. We've also done work with thiol-maleimide cross-linking, thiol-thiol cross-linking, using ketones instead of aldehydes to make the hydrazone bond. And these all modify the stability of that degradable bond. So we can sort of tune things from, something that will degrade in a day or two. And in other cases, combining zwitterionic hydrogels with some of these cross-links, we can get things to stick in the body for up to 6 months before there's substantial degradation. So I think that's a benefit. It's traditionally viewed to be a negative of synthetic hydrogels. How do you get rid of them? And I think, it's not that there's no concern in that. That it's we have some data that we are pretty convinced it can clear through the kidney, but it's a fair question. But I think the flip side of that is the control you can get using a synthetic hydrogel with a particular functional group linking those together is much, much higher than you can achieve with some other approaches. So I think that there's a trade-off, but I think we at least think we're on the right side of that trade-off.
Okay, great. Yeah, indeed, it's always a balance that we are trying to achieve. And then talking about this, we know that it's very interdisciplinary, this field. We talk about materials, chemistry, biology, physiology. And on this topic, how do you work with this interdisciplinarity? Do you have people in your group from different backgrounds, or you count more on external collaborators? How is this in your group?
I think it's a combination. So I have people with biomedical engineering backgrounds, chemical engineering, chemistry, biochemistry, life sciences, and nanotechnology engineering too. So those are sort of the undergrads of my graduate students. So we try to get some range of expertise in the group, and some of the postdocs that we hire are sort of targeted in that way too, right? So here's something I don't know as much about, so find someone who does and then try to help. But I mean, I think it's very difficult in a biomedical application to have one group know enough to do something really practical, right? So we collaborate with a lot of colleagues, and I think we're really fortunate at McMaster to have a really strong medical school and health sciences program. So there's a lot of colleagues that are available to make those collaborations. So I just off the top of my head, I mean, we have collaborators in pathology, psychology and behavioral sciences, internal medicine, respirology. So a range of disciplines where, there's been some sort of problem that they've identified, or we've identified them as, would this work for this application? Well, let's find someone and ask, and made a collaboration and, maybe co-supervise a graduate student or something like that. So I think that's the only way to really do impactful research in this area. I think if you try to do it all alone, you're going to make a stupid mistake just because you're not going to know something important.
Yes, for sure. We all have a complementary knowledge on the topic. And in the context of tissue engineering is still, which are the characterization techniques or assays that you and your group normally or mostly perform in your samples?
Sure. So, I mean, we work almost exclusively with hydrogels in the tissue engineering space. So we're always worried about swelling as one thing. And swelling can be a good thing or a bad thing, depending on the application, right? So if you're trying to fill a particular defect, it can actually be very helpful to make sure that the defect is completely filled, right, with something that's easy to administer. Whereas in other cases, if you swell, you might create pressure in an area that you don't want to create pressure in. So depending on the application, that's something we worry about. We also think about mechanics, obviously, as the mechanics of a tissue scaffold are really critical for how successful it is. That's unlike some of the other applications. I mean, in drug delivery, do we care about the mechanics of the implant? As long as it's stable, we probably don't. Whereas in tissue engineering, cells are going to sense that, and the native tissue is going to sense that. It can really dramatically change the biological responses, both to the cells you're trying to deliver and for the scaffold to the outside biology. So we spend a lot of time and effort thinking about that. And then we try to do, I would say we're not experts ourselves in cell biology by any stretch, but we typically do things like live dead assays, stains for extracellular matrix production, total DNA counts to see if our cells are actually, if we're proliferating cells inside the scaffold or if they're just kind of hanging out. Things like that. So that can be quite challenging because staining cells inside a scaffold, it's very hard to get the cell stained and the scaffold not stained often. So there's a lot of challenges with characterization. I think that's actually a key challenge in the field. Everyone does something a little bit different. So to compare paper 1 versus paper 2 can be really difficult because there's not a lot of standard methods that people use to characterize that. But that's typically what we do, some sort of swelling measurement, a mechanical measurement, and then what are cells doing in terms of, are they viable, are they growing, are they spreading, things like that.
Yeah, and on this point about a standard method, I think also the mechanical properties is something that we see a lot of variation in the literature when we talk about mechanical properties and hydrogels. And in this context, how do we work to assess the mechanical properties of your hydrogels? Which methods normally you use?
So it's a great question. And I think one of the challenges is for a cell, I mean, most of our mechanical methods are bulk methods, right? So we're compressing it. So we do sort of micromechanical tests to compress. We have an instrument that you can sort of put a little rake in and pull sort of thin tissue scaffolds. So that gives us an idea of tensile strength, which is very, very helpful. Rheometry, you can get some idea about the shear response of these materials, which may or may not be relevant depending on the application. But I think one of the key questions is always, what does that number mean, right? So, if my question is, can I pick it up? Can I pull it? Will it stretch? Will it break? I think those are meaningful numbers. But if the question is, what mechanics is the cell sensing? And how does that impact the cell performance? It's a very different question because in a typical tissue scaffold, you have a lot of pores. So your bulk mechanical property is going to be a result of your porosity plus your gel. Whereas the cell doesn't really care about the porosity, it cares with what it's interacting with, which is the actual hydrogel itself. So to get some insight into that, we usually do bulk hydrogel measurements as well as scaffold hydrogel measurements to sort of get an idea of what the gel might look like and what the scaffold might look like, incorporating the pores, and try to put those things together. And there's ways to locally probe hydrogel mechanics. So we've tried AFM, but anyone who's tried AFM in buffer knows that that's an extremely challenging experiment. So I think that's a key challenge. But it again, and the other reason it's challenging too is even if we do the bulk hydrogel, the mixing of our precursor polymers doesn't necessarily happen the same way in that context as it does when we try to make a scaffold, where the mixing might be very different. So relating that result back to the scaffold is even harder. So I think there's a lot of challenges in mechanical characterization. I think having as many tools as possible and subjecting your material to as many tools as possible to get different data points that they then sort of try to put together to get a comprehensive answer to those questions is really important in that context.
Yeah, I think you mentioned a nice point. I remember in the beginning of my PhD, I worked in a lab where they have also a group that work with metals, and it was for tissue engineering. And I always heard them telling about improving the mechanical properties of stents or any device that was made by metal. But I always thought metal are already so strong that why is this question, like how pertinent is this question in thinking about the application itself, right? So we always get many different parameters from many different tests, which can be great, they can complement, but we should not forget about the application, about our aim, how this parameter correlates to the application itself. I think it's
Absolutely. And particularly when there's cells involved, right? It's one thing if
yes.
it's a biomaterial, it's a lot easier to make those interpretations. But when there's cells involved, I mean, if you're trying to regenerate cartilage versus you're trying to regenerate neural tissue, right? There's a huge gradient in mechanics you need to be worried about. And that's, again, more the local mechanics of the material itself rather than the global mechanics of the scaffold. So I think it's a really, I don't think there's a satisfactory answer out there right now, at least that I've seen. And then, this is what everyone should do to measure the mechanics, and this is what that number means. So I think it's an area the field in general has to think a bit more about in order to really get consistent and comparable results.
Yeah, so I think I will move a little bit. Is it still on science, but maybe going a little bit out of biomaterials? Thinking about this context of pandemic. You mentioned, I think, that you were involved a bit in projects related to COVID.
Oh, that's right. So there's been a couple of COVID projects we've been engaged in. One is, as I said, the anti-infective surfaces work. So I don't know if you've seen these commercials for Microban on TV. So it's I think it was a product that I think Procter & Gamble makes it, but they had it just at the beginning of COVID. I think they're releasing it for other things, but they got super lucky and sold a lot of it. But if you've used it, it's actually horrible. So it dries really, really slowly. It's kind of sticky. It's really unpleasant. So we've been working to try to develop some better methods that give you that not only 24-hour protection against bacterial transfer, but maybe even longer protection, but that dry a lot faster, that use materials that are a little bit less problematic, and something that hopefully people would rebuy. So that's the project we've been doing a lot of work on, and not only for consumer applications, which I mean with COVID, transit rails and countertops has sort of been hot, right? But thinking about things like bioreactors, it's biofilm information is a huge issue. So if we can make a stable enough coating that can last for several fermentations, that would be very beneficial. And the other project we've been working on is sort of inhalable microparticles for treating lung fibrosis. So again, this is a project we were working on before COVID, as we were trying to make these very, very porous, light microparticles that are easy to inhale, can get into the lung, and actually are inherently immunomodulatory. So they help to sort of reprogram macrophages from the fibrotic phenotype, which sounds nice and healing in wound healing context, but in fibrosis are bad, back to a less fibrotic nature. So we've been looking at how to leverage those particles for treating COVID patients, sort of the end stage COVID patients, where fibrosis of the lung is sort of what ultimately does them in when they just can't breathe after these cytokine storms and other sorts of immune responses that a lot of these patients get. So trying to prevent that is using the polymer itself as a therapeutic is something else we've been working on.
Okay, thank you so much, Todd. I would say, Kathleen, do you have still some questions in the science block?
No, you've covered that very nicely. Thank you very much. We would like to continue a little bit more on the professional side of things. your professional life in the lab, like what is a day in the life of Todd Hoare in maybe there's two scenarios that we would ask that,
Please.
right? Because data life in the lab, during COVID versus what would be a typical day, that pre-COVID, or when we had a little bit more leniency when it came to being in the lab. So what would be a typical day in those two scenarios during COVID versus non-COVID?
Well, during COVID, I'm sitting in my windowless basement, staring at a computer for 8 hours a day, but that's pretty much everybody. So I don't, that's probably not too interesting. And pre-COVID, I mean, I think with most professors, particularly with those with larger research groups, I mean, I don't work in the lab myself. I think my students would be upset if I touched their instruments because I'd probably break them at this point. I think there's only one instrument in my entire lab I actually know how to use. And it's actually kind of embarrassing. I won an award a couple of years ago and I had to do a science fair on Parliament Hill. So I brought some of our thermal response to do a demonstration for the MPs. To do that, you need a hot plate in order to heat them up. So it's like, right. Okay, grab a hot plate from the lab. Spent about 5 minutes trying to figure out how to turn it on. So thankfully I figured it out, but it was kind of embarrassing. So I mean, I don't do a lot of hands-on research anymore. But what I did like to do when I was in the lab is just sort of walk through and see what people are doing, ask questions. I think probably the most fun I had in any week is on Friday mornings, we have a little kitchenette outside our lab. I would just go and sit in the kitchenette with my computer. And if someone came up and talked to me, that was great, but they didn't have to. But just the sort of the random conversations about science, or, a couple of people would come up together and it's like, I have this idea, let's talk about it. That was a really, really fun thing to do. Harder to do online. It's just, you can't be quite as spontaneous, but
yeah,
that was definitely my favorite part of the week.
Yeah, it's true. The lack of the casual interaction with humans does make for this time to be a little bit more challenging. You almost need to provoke these meetings to make that possible. So I guess one of the things that I was thinking about is, what would you tell your younger self about this field? Like, you would sit down with your younger self. What would you say? Or what would you recommend? What advice would you give yourself?
Yeah, I think my advice would be less conservative. As I think as a young faculty member, you're kind of paranoid about generating papers and making sure everything works, right? And you're very paranoid with money too, as you have a limited amount of money, so you're not sure, should I hire another graduate student or should I save this? But I think what I've learned is, it's probably a lesson I should have known before, but you have to invest to make the next money too, and that's true in scientific discovery as well. I mean, you have to sort of try something hard, and if it doesn't work, that's okay. But you'll probably learn something interesting along the way. So one, entire stream of projects in my lab actually came from something that didn't work for another project, but we saw something interesting. It's like, wait a minute, we should actually try that, and it's led to several papers. So I think being less scared to try things that are maybe riskier, whether that's investing or in terms of just doing ideas in the lab, that's what I would say. But I think that's easier to say now as a more senior person. I guess I'm a senior person now. It's kind of a weird, but
hindsight is 2020, right?
Exactly.
So, yeah. Well, but it's nice to be able to transmit that onto others, and I guess for those, what would you suggest to those that are listening, the students that are listening to this interview and who are starting off in the biomaterial world? What would you have as suggestions to them?
Yeah, for students, I think the important thing is to just try as many things as you can, right? Because it's biomedical engineering is such a giant field, and there's aspects of biomedical engineering I am completely uninterested in. And if I happened to have stumbled into those in my undergrad as opposed to the ones I did on more on the materials side, I don't think I would have been in the field. So I think it's a good idea to take any opportunity you can get to get a bit of exposure to different areas and subfields in the area. That could be, asking for summer internships, volunteer positions, attending conferences. There's so many good student conferences. Actually, that's probably the one good thing that's come out of COVID, all these online, very accessible virtual conferences and workshops that you can just drop in for free or a very small amount of money to attend and learn things. So I think it's just exposing yourself to as many ideas and concepts as possible, then sort of evaluating after that, okay, did I like that? Do I want to learn more about it? Or what part of that did I like? Or do I never want to speak of this topic again and go on to something else? And I think all those are good outcomes, right? So I think it's just delving in and getting as much experience and exposure as you can.
Interesting. I'm sure that's going to be passed on. So as a leader working with these students, working with your team, what is it that, I mean, and potentially this is a two type of question during COVID versus non-COVID. How do you go about motivating your team, your group, your teams, I should say, because you're clearly working with multiple groups. How do you go about motivating them? How do you get them excited about the projects that they're working on?
I think it's difficult to excite people who aren't excited at the start, right? So I think the number one thing is to hire people who are really engaged in these ideas and making these things work. Because if you don't have the people in place, I think a leader can only do so much to motivate them. But in terms of trying to keep, particularly in COVID, it's difficult, right? We can't meet. We used to have a lot of food. So I actually have one of those waffle makers that you see in hotels. So I bring the waffle maker to group meeting in the morning, and we just make waffles the entire time and talk science. I mean, those sort of things are good for team building and just getting to know people on different levels. So we do a canoe trip most summers, which we did manage to do this summer, which was good. Down the Grand River, which is nearby. So having those team building activities is, I think, good. And also celebrating success, because I think research, as I said, is a lot of failure, right? Just thinking back to one project in my postdoc, I needed a microgel that did something very specific, and the one that worked was number 87. There was a lot of failure, but then there was a big success when we figured that out. So I think making sure that those successes are celebrated. So highlighting people on social media, or when we could have food, having some sort of celebration like that. I think it's motivating. But I think, as I said, the number one thing is to pick people who are motivated and have a team that is enthusiastic about what they're doing. And then in research, I don't know if there's a substitute for that.
That's true. Passion and interest and drive are part and parcel of people's personality, and that no matter the field happens to add a lot of value to achieving specific goals. So I guess how do you go about attracting talent to your teams? Because I can imagine COVID has made that a little bit more challenging because you're not at conferences. You're not really meeting with people. How do you go about doing that in these times?
I mean, that's a great question. I wish I knew the answer to that. But I think it's something a lot of people are struggling with now. So I think there's a couple things you can do. One is I try to participate in a lot of professional societies. So I've been in the leadership of a few Canadian societies, and I think that's really good to make sure that you meet students through that, and you also meet other professors. So, just realistically with grad student decisions in particular, most undergrad students would go to a professor they trust and say, well, that's what I did. I'm kind of interested in this. Who should I email, right?
Yeah.
So the more professors in the area that the more professors might say, oh, you should email Todd. It might be something that you're interested in. So I think that's been very valuable. And also, I mean, a lot of people are engaged on social media, so I keep a Twitter page, which I don't know if I keep particularly well, but just having that out there is actually made a couple of leads that I might not have had, and also on LinkedIn, I've actually gotten 2 research grants through LinkedIn, where
Nice.
people have searched and found expertise and contacted me, and then we ended up doing something. So I think that sort of keeping as visible as possible as you can during these lockdown-ish times is good, and however you can build a network to do that is beneficial. But I wouldn't say I have a magic bullet to that. It's certainly been much harder to recruit.
Yeah, it is. It is much. Yeah, it is more challenging. So I guess some of these organizations or societies that you were talking about. Which ones are you part of?
So I've for a long time I've been engaged with the Canadian Biomaterials Society, which I think is a really nice cross section of network of researchers in different areas of that field across Canada. I've been a past president of the Controlled Release Society, the Canadian chapter, and now I'm the chair of the Macromolecular Science and Engineering division of the Canadian Institute of Chemistry, so more on the polymer side. So yeah, it's harder too when your research sort of spans different areas to sort of know where to, what community you want to be part of, or sort of in between a few. But I think those have been the societies I've really gotten a lot out of, and built some nice, good collaborative or just friendship relationships with other colleagues, and then and they've helped to make sure at least students are aware of what you're doing, and then everything from that is sort of up to the interview, and up to how convincing I am for really good students to come work with us.
Speaking of which, how do you recruit students or collaborate with PIs, or what are the most important qualities that you seek in these different groups?
So I haven't found a huge correlation between marks and excellence in research, to be honest with you. I mean, I think there's a baseline level of academic performance you have to have, but I would say probably my two best early PhD students came in with B+ averages. But they just were incredibly enthusiastic, very driven, very committed, and made it happen. And I think that's really what I'm looking for when I interview people. Like, do you have the fire in your belly to be a researcher? Because I think it's very different than being an undergraduate, where you're writing tests and you're doing design projects, and in theory, to go into the lab and execute and deal with the failure, and keep focused with deadlines that are sort of vague or non-existent, right? It's a really different skill set.
Yeah.
So it's really, I think, the enthusiasm, the drive, and also just being a pleasant person to work with. I think, particularly when you have a larger group, there's nothing worse than bringing someone in who's not pleasant. So I think.
I would agree. It's definitely one of the questions that all of us should ask when we're interviewing or meeting people. Do I want to work with this person? Right? Like it's definitely a significant, or it plays an important role. Let's just say that, right?
Absolutely.
And what about your collaborations with PIs? What, other than the actual subject that they're working on, are you looking for any particular, this might be a good venue, like, are you looking for any particular collaborations right now, or what do you look for in working with other PIs?
I think complementary of expertise, right? So there has to be a reason to collaborate, as opposed to just kind of, let's do something together that's sort of vague, right? So typically a lot of our collaborations are in areas I know little or nothing about, and then that person sort of fills in the gap. But I think a lot of the same criteria apply, right? I think you want to be working with people who share your values in general. So in terms of research ethics, in terms of treating students in the right way, is if you end up being in a co-supervised relationship with another PI supervising a student, and they have different expectations, different demands, different ways of supervising, it can lead to challenges. So I think looking at sort of obviously the scientific fit, but also the personal fit in terms of does that work on a personal level is important. And we're always interested in collaborations. As I said, it's usually, occasionally it's us saying, hey, this is a cool property, can we use this? But usually it's us being completely with no clue about why someone might use it, and just kind of out there presenting, and someone comes, hey, wait a minute, I might be able to use this in this application, and seeding collaborations that way. it'd be great. So if anyone listening has heard something about, you know, nanogels or injectable gels and thought, hey, I could use that in my research, I mean, we're definitely happy to chat.
We'll make sure that that communication takes place. So looking at the future and looking at vision of this industry, what are the emerging challenges and trends in the field that you perceive?
I think in tissue engineering right now, there's so much focus on 3D printing. And I'll have to admit, I'm a slight skeptic about 3D printing, although even though we're doing it. But I think, but I think the challenge is the resolution, right? So to get down to the resolution of the printed features that actually mimics some of those features in native extracellular matrix that cells use to spread and signal and communicate. I think it's a challenge in the 3D printing area, particularly for something that's scalable, right? So some of the techniques that are very precise, like stereolithography, you know, they might be able to get down to that resolution, but to get a big tissue from that process is extremely slow. So I think there's a lot of technical developments there that it's part in around the printers, but also in terms of the inks. So what types of hydrogels or starting materials can you use that actually can enable some of those properties to happen? And right now it's a very short catalog, I would say, of inks that are typically used in bioprinting, and that's where we feel is the opportunity in the field, and where we're trying to work on filling that in and making some, you know, expanding the catalog to give different options and properties there. And I think the other challenge I would say is getting, you know, we've had reasonable success, I think, making simpler tissues, right, where there's maybe one cell in them. But it's been much harder to make more complex tissues, because you have to have the cells in the right place, you have to have them communicating with each other in the right way, propagating in the way, you know, relative right way that you need. So I think that's what's motivated a lot of our work, trying to load scaffolds at the same time as we make them. So hopefully we can get a bit more spatial control over where cells are and how those cells interact with each other, and mimic some of those native biology features a little bit better to maybe aim toward a bit more complex tissues in the future. But those are the two challenges that come to mind.
Demetria, did you have anything that you wanted to follow up on this particular question?
Yeah, Todd, do you think by the beginning of your answer, I felt, do you think that maybe we are focusing too much on 3D printing? And for example, injectable hydrogels, for sometimes we don't really need to print. So would you think that we are concentrating too much on just one avenue?
I mean, I'm going to give a completely biased answer, but personally, I think yes. I think it's maybe a little bit. I mean, 3D printing does solve the problem of putting cells in the right place and putting cells in the matrix at the same time. So I think one of those two problems identified, 3D printing actually addresses really well. But it doesn't address the invasiveness issue. Although there are some handheld 3D printers that for sort of open surgeries people are using, which I think that is actually quite interesting. And it also doesn't really address the structure issue. So how do you make those, you know, hundreds of nanometer features like some of those native fibers we see in the ECM that are really important for tissues to grow? How do you actually reproduce those? So I think, you know, there's definitely value in it. I certainly don't want to say there isn't, but I think thinking about other approaches that would enable much less invasive delivery and reproduce some of the features of native extracellular matrix on the scale, the length scale that they're present in real tissues, I think it would be worth some second thoughts on other ways to do that, and that's some of our motivation, at least. Hopefully we're right on that. I guess we'll find out. But I think there's a lot of, you know, it's a multifaceted problem, and 3D printing addresses a couple of those problems, but I think it might struggle to address all of them.
I feel like you've answered this to a certain extent. My next question, which is more specifically in this field, what do you know, what do you see in the next 5 to 10 years? And I feel like you sort of like, let's be a little bit more targeted. Like, what do you see in the next 5 to 10 years in in these specific applications or this specific field?
Yeah, I would say two things. One is, as I said, trying to make a bit more complex things, right? So being able to incorporate cells, the different types of cells, in ways that they can interact in a way that we can make more complex tissues.
Mm.
I think we're starting to get there, and I think that's an exciting area. I think the other really interesting area is sort of being able to do this in the operating room. So there's some papers I've seen that I really think are interesting with handheld 3D printers that can do gels that a surgeon could actually just do on site, or some handheld electrospinning equipment is available now, so you can actually make those nanofibers, but put it right on a tissue in an operating theater. So just the thinking about avoiding sterility issues, things like that, and filling the defect that you actually want to fill. I think those are some really interesting applications to sort of put this technology, so it's not just in the hands of engineers, but put it in the hands of clinicians to see what they can do with it, and then I think getting that feedback, this would be good to have, this is useless. I think that would be really beneficial to making these things a lot more applicable in the real clinic, which I think we think about a lot. We think about commercialization a lot, so I think that's one thing. I think the field is moving that way, I think, and I think it'll be really interesting to see what happens when that does become more common.
So I guess my last question is, more on, like, if you had a magic wand and you can get whatever you wish for, what would you actually wish for in this field?
Uh,
In
can it
this
be a
field,
COVID
because I
related
mean,
wish?
we would love a magic wand on many other fronts because we get rid of COVID, right? But putting that aside and looking at this specific field, if you had a magic wand that you could whip up and use, abracadabra, what would you wish for?
I guess I could give a technical interview, or answer to that. But I think the thing that would help push it through the most is if clinicians were far more engaged with academic researchers. And I
Mhm.
understand there's some challenges with that from their perspective, particularly right now. They're under so many demands, and then their pay structure doesn't really reward spending the time working with researchers to solve problems. But I found in our research, and I think this is fairly common, it's really easy to engage the preclinical health sciences people because they're very interested in solving these problems, and we can offer technology to help them solve issues they've been working on. It's much harder to engage with actually people and clinicians who are dealing with patients directly. And I think having that knowledge in those conversations early as possible can avoid a lot of the mistakes that researchers often make in designing things that ultimately don't become clinically relevant. So I guess if I had to pick one thing that's sort of on the technical side, but not breaking out of my basement, but on the technical side, I think just increasing that engagement and finding ways to really create some mutual benefits, I guess, in that relationship, that surgeons or clinicians can see the benefit of working with fundamental researchers, because the time scale is so long on a lot of these biomedical things. I think it's hard to make the value proposition sometimes to very busy people, but
Yeah.
it'd be really nice if that was able to be done more efficiently. I think we'd get to solutions to some of these problems a lot faster.
Right. So there's definitely a disconnect between those two worlds that you'd like to bring together, so to speak.
Ideally, yes. So
Yeah,
hopefully that would
that makes
happen.
sense. That definitely makes sense. So thank you so much, Dr. Todd Hoare, and joining us today in the Expert Corner. We certainly look forward to what's coming ahead for you and your teams. Thank you for being with us today.
Thank you. My pleasure.